Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, welcome back. Here we are again with lessons from biology under Unit 3 like we said. And like I've already mentioned, we are still in Unit 3 and this should be an advantage for the senior sixes because you take this as your review. We are looking at now describing the structure of the DNA and at the same time to be able to see how within the structure of the DNA the bases have to pair up. So we refer to it as complementary base pairing. That's our lesson today. We should be in position to cover one, clearly describing the structure of the DNA. Two, to be able to describe how complementary base pairing occurs so as to be in position to form something of this type. Now, remember, in the other homework, we looked at an outline of the structure of the DNA, and we also looked at the whole structure within the chromosomes, and part of the homework was for you to describe. Now, here we come again. When we use the word describe, what exactly are we looking at? How does it differ from outline? If we looked at outline in the previous lesson, and here the homework was for you to describe, I gave you a hint as a ladder. Could it be the reason as to why finally in our image here, we have a structure like this? Now, let's look at this. We need to be very clear about description. That's why the word describing has been underlined. Two, we also describe how. So the how would take us through the process. Description is a step-by-step event if it is a process. It can as well mean that if there is a structure you're looking at, what are the parts that make this structure up? That's one. What are these structures? How are they arranged? And what is the overall shape? This is what we expect to capture in the DNA structure if you're to describe it. So, the structure of the DNA, excuse me if I cannot to put on my glasses, be able to read very well. You have one composed of 4 nucleotides. Last time we looked at a nucleotide, the smallest unit, the small part out of all this structure. Remember, last time we said if we are to look at this, then this is our nucleotide, one nucleotide. So they're saying one, it's composed of 4 nucleotides forming infinite chains with greater variety. That's a very interesting statement. Non-ending, long chains, so they keep adding up to build a long structure. So a polynucleotide, like we said. 2, when we talk of greater variety, it now takes us to something we are going to look at in lessons to come on afterwards, where we are looking at what is composed, what is contained in these structures. How is the arrangement? If it is infinite, how much information is it containing? How is the variety looked at? So if you so much stored in, and then you have a lot of it, different forms then, could it be taking us to something to do with so much information stored, and so a variety of information forming different types of genes like we looked at last time? 2, that each nucleotide has, 1, a base, which we looked at as an organic base last time. 2, a phosphate group. And 3, a sugar. Remember, not the same table sugar you use at home, but this we said should be a 5-carbon sugar. So we call it a pentose. And remember, Based on the structures, then we shall look at this as a deoxyribose sugar. The base here, we have a lot of options. This can be any of the four. So here we go. Any of the four. We looked at these last time, where we were looking at the small ones and the large ones. So it means, therefore, that each nucleotide is going to have this, that, and that. So you have this, that, and that. Then you have this, that, and that. So we are going to have a phosphate. Then you have your sugar here. Then you have your base. The same applies to this. But where are the differences coming from? If we say only 4 types of them, then we are going to look at 1, 2, 3, 4. And when you look at the structures, each of the 4 bases, one is different from the other. Second observation, you would see that you have this and this more or less the same size in terms of the structure design, and this one more or less the same. So comparing this with that, then this is small. This and that, then this is what? This is small, this is large. Now, when we go straight to the composition in the description, based on how we have said we need to use the word describing, does the statement capture a description? If a learner said that, for me, I look at the DNA as a ladder. If I go to my illustration here, I have a building. So this is a window, this is a door, so this is the wall. So if I'm to climb up here, then I need to use the ladder. That's one. So if I say that the DNA molecule we looked at last time is in form of a ladder, as you can see here, then I need to look at, if I have the ladder, what are the things that make the uprights of the ladder? So here I go, the stands. 2, what are the things that make the crossbands of the ladder? So the crossings where a person will step on as he climbs. So if I say that the DNA consists of 2 strands, then our DNA would be compared with a ladder. So this is a strand, this is another one, arranged in the form of a ladder. So 1, the ladder has 2 components, the uprights and the crossings. So we need to check, transfer this to here, and then ask ourselves, if I was to move this here, then this would mean that this straight part would represent this part. So if I use a marker here, then this would take me from this part to represent this section, and this part to represent the other section. The crossings, which we have here, composed of organic nitrogen bases in form of a pair. So this and that will form a pair, this and that will form a pair. So this is our ladder finally. That the 2 strands then coil around themselves in form of a spiral. So like twist, like the way you have your fingers turned round, or you have your arm folding on another, or the way you have threads, a thread woven around another one to form a spiral lining. So you have a helical shape as another way of expressing it. And therefore, at the end of the day, we have it as a double strand, which takes us back to our primary objective. Can we tell how the shape of the DNA is designed? Now, when we talk of complementary nitrogen bases, if we are to have a structure like this, then we have a lot of things bubbling in our heads. One, how are these bases paired? Is it by random design? Do we have a possibility of following on the structures? Is it also possible that maybe there could be certain forces that hold them together? And if these forces are holding them together, are they always permanent? Are there situations where they are going to separate, like the way I'm removing my hands, the fingers separating from others, or they permanently remain interlocked? Which takes us to this wonderful and interesting part. If I have my structure here, and I have another one here, these two are not the same. This is curved inwards, this is curved outwards. It therefore means that this would lock in here. Now, the locking, I want you to follow very carefully here, does not mean that this fist is having the same shape as this. In the same way, we would not say our feet have the same shape as shoes. Think about it. Shoes and the feet, do they have the same shape, or one locks into the other, and so they fit into each other? Which one would actually be bigger? Is it the shoe or the foot? Now, which takes us to this interesting section of what we need to be having in our mind, bubbling in our heads, and wondering, how does it all fit in? Are they glued together? Do they separate? If you can remember way backwards when you're studying about cell division, even if it was senior 1 or senior 2, where cells multiply, but every time they multiply, the content also has to first multiply before we can distribute it to these cells. So if you had a DNA like this, how is it going to make its own copies to be a lot? Yeah, so it takes us to this. Can we break this apart, separate this component from this, so at the end of the day, we have more copies coming in, which we shall also look at in the next components of replication, making copies of the DNA, making copies of the information for new cells. Now, so here we come. For these bases to come together, we look at how the process of pairing occurs, and so we use the words base pair complementation or complementary base pairing. But what is behind this pairing? So pay attention and look at this so very carefully. I have box files that are run in the office. These box files have a shape that you look at here. This side is relatively bigger, and this one is small. So if I hold it this way, this is big. If I spin it around, this is small. So it is the same design as we are having here. Now I want you to do the same thing back home where you are. Find anything that is having multiple arrangements, and then you look at it. If they are your small books, try to arrange them in a way and see when they actually fall, that they don't remain stable. So here you go. Take a look. One, put the second. I'm trying to arrange my office. Put the third, trying to arrange my office. Then put the fourth. Looking at this, anyone would come and ask, how do you arrange your desk like that? How do you arrange your shelves this way? Maybe I would have to think, what if I arrange them the other way. So I have one here, and I put another one. I'll get this, and I add another one. Boom. So there we go. That's my arrangement. How is it? If I was to make measurements from here to here, how much of the measurement do I have? You should be able to check that very carefully. Simple estimates. If I was to make the same arrangement here, how far have I deviated from the answer? A simple conclusion would be that if I was to put a ruler on top, I more or less would have a horizontal straight section as well as the bottom, as you can see. Which takes us to the arrangement of the DNA. In complementary base pairing, we do have one. It refers to the way bases can bond to form two nucleotide chains. So I would assume that in the middle here, this is one section of the base, another section of the base. So one base on this side, another base on that side, somewhere in the middle, to be able to have such an arrangement. Meaning that at the end of the day, my shape is clear. 2, the arrangement of the nucleotides on one chain determines the arrangement on the other. What does this mean? That the way I first had my files, the way this looks should determine how this should come and fix in. The way the second on the same side on the left looks would have to determine how this would fit in to be able to lock and have our uniform structures you can see. I think it makes sense now. A large base to do a small one here. So the locking should give us something of the sort. 2, there has to be an antiparallel arrangement. What does this mean when we say antiparallel? So we go math. You will look at this. I'm using this component for the activity for you to be able to understand that this faces up. So we go up. And according to the shape and design here, this is facing down. So if we were to arrange these and give the numbers, then we are having number 1, number 2, number 3, number 4. Carbon number 5 is located somewhere here. So this goes up. 1, 2, 3, 4, 5. These numbers represent the positions of the carbon atoms on the sugar, the deoxyribose sugar. When we come here, the arrangement now goes down this way. So you have this going down once, while this is going up. That's what we mean when we say antiparallel. Next, in the process of base pairing, so we are still moving on. In the process of base pairing here, you have 2 categories of the 4 bases we were looking at the beginning. That you have the purines, which are the large bases. We looked at them in the previous lesson, so you need to remember. And the pyrimidines. These, we refer to them as large bases. Try to recall the previous lesson. And these are small. Now, in pairing, we look at a large base pairing with a small one. In the process of pairing, we do have the bonds that hold them together. So here we go. Simple rule. A large base A will always pair with a small base T. A large base G would always pair with a small base C. So base C pairs with G, base T pairs with A. A is a large base, G is a large base. C is a small base, and so is T. So in base pairing, the rule is that A goes with T and G with C, not the reverse. Which takes us to one of the activities here. Now, I want you to pay very much attention here and carefully focus on this. I'm giving you only 15 seconds to look at what I've written here and wear it here on the activity before we do something. So focus there and see what we mean when we were looking at our boxes. And now from there, here we come. Are we able to do this activity? If you're close to your TV set, you should be able to get close quickly, maybe take a snapshot using your camera or phone and pick it out and see how you can work around it. How far? So let's do it. I'm very sure you're done now. We are going to transfer the concept we have just put here using this into the activity and do it, and then we are good to go. If we start from the center, so you have C, you also have A, you have B here, then F, and then thymine. They want us to give the names of the parts labeled. Remember, we have to keep focusing on what we are looking at on the box files. So here we go. If we look at A, and then B, and then C, then F, and then this one is red labeled for us. Now, in our attempt, we need to ask ourselves, why has this structure been given a label? So here we go. Let's do the labeling. So we shall have our phosphate group. Then we shall have our 5 carbon. Remember, this is DNA, and so we have to write it as deoxy. Ribo, sugar. Remember, it is a 5 carbon. This is how we count it. 1, 2, 3, 4, and 5 before we go to the phosphate. There is a bond between a phosphate and another carbon sugar here. Another carbon atom on the sugar. F would be given its name depending on thymine that is labeled for us, which takes us to this statement. Where is it? Here. That the sequence of nucleotides on one chain, this is our chain, is going to be dependent on the sequence of nucleotides on the other. So if therefore in assessment one is named for you, you should base on the rule of pairing to be able to know how the other one can be named. So we move from where thymine is here, and we come to our simple rule and check where thymine is. T, which was given this full name, falls here. So it means, therefore, that the member of the pair would be this. So I write adenine. Remember, it is a small base pairing with a large one. Back to this structure that has to keep this same arrangement and stable takes us to how these are arranged. Same arrangement here that is wired here, which now takes us to how we've been able to look at our second objective, describe how complementary base pairing occurs in formation of a double helix, double, two strands. So you have this and that. This is what we call a double-stranded DNA structure. So sum it up, we have one, we've been able to describe the structure of the DNA, one being looked at in form of a ladder by simple description, composed of uprights and then the crossings of the nitrogen bases. That the two coil up together around each other to form a helical shape and so appears double-stranded, as you can see here. Pairing, complementary base pairing, as we do have, the rule still remains the same. A pairs with T, G with C, as we have in the structure here. And I want to reserve this time for you to have some bit of activity to do, which you will be in position to later try on the later time. So here you have two forms of homeworks you're going to do that are going to be wired to you so that you have time to capture them. So how do we do this? We are having this. We can cover this. We've done it. That's one. You should be capturing that so quickly. And this. Oh, sorry, I'm covering that. It doesn't have to be that way. So, yeah. So here you go. So this is your number one, and this is your number two. Capture that so very well and see. I think we might not need this at the moment, such that you are able to fully focus on that. Take a caption. If we read through quickly, you should be able to check. In an organism, 26% of the bases in DNA are found to be A, adenine. What percentage would be for C? Show your working. Here you have a table of data. After looking at it very carefully, you should be in position to, one, how do the results relate to DNA structure, especially the model? And you should also be in position to look. Now, here is the hint. Is the DNA double-stranded? Is it for humans? If we have E. coli as bacteria, then wheat and sheep and others, go back to your senior one and check, how do I rate these numbers with complementary base pairing and the question here. And with that, I think we come to the end of the session today, and I'm pleased for you to have been able to follow very well, capture the objectives, and be able to run through very, very, very carefully. And thank you for that today. We meet in the next session after this. Thank you.